I'm Monica Bartus, and my experiment was on preventing and reducing acidification in freshwater. I wanted to know how plants and lime affect acidity levels. First, I had to do a bit of research. I needed to know how acidification worked and how lime and plants would affect it. As carbon dioxide is absorbed by water, a chemical reaction occurs. Carbon dioxide, water, and a carbonate ion combine into two bicarbonate ions. This results in a higher concentration of hydrogen ions and a decrease in pH. One way to reverse acidity is to add a base. For example, when soil is too acidic, farmers will add lime to neutralize acidity. Another way to reverse acidity is to limit carbon dioxide from increasing the amount of hydrogen ions. Plants use carbon dioxide in photosynthesis and then expel oxygen. If the plants use the carbon before the chemical reaction occurs, the water won't acidify. Oceans are helpful to prevent climate change. They are a huge carbon sink that absorbs 22 million tons of CO2 per day, but we are overloading them. They have increased acidification by 25% since the pre-industrial era. Many organisms are sensitive to changes in pH. It can injure fish or make them use energy to keep their body chemistry normal. Shell and reef-building marine life need carbonate to build. Many of their shells will also dissolve because of the higher acidity. As the research showed a prevention of acidification from the lime in plants, it was hypothesized that both would reduce the acidification of water, and it would reverse already acidified water. It was also predicted that the lime would prevent the most originally, but as the plants cleared CO2, they would catch up. Then, experimentation was started. The presence of lime in plants were used as the independent variables. This was the control, these were plant only, these were lime only, and these had both lime and plants. Espoma garden lime was used for pelletized lime. Java ferns were the plant in the experiment, and the pH was measured with a Dr. Meter pH meter, which accounted for changes in temperature. 20 32-ounce jars were filled with 350 milliliters of distilled water. 5 jars were set aside to be the control group. For the lime group, 2 grams of pelletized lime were added to each of the 5 jars of water and stirred. For the plant group, a single plant was placed into each of 5 jars and the leaves of each of the plants were tucked under water. For the group with both lime and plants, 2 grams of lime and a plant were added to another 5 jars. The jars were exposed to moderate sunlight for an hour to help photosynthesis occur. An adult supervisor was then asked to shave off dry ice to be scooped and measured. The supervisor needed to be present during experimentation to refill the dry ice as it sublimated. The initial experiment tested water acidification following a single dose of carbon dioxide. Following the initial measurements, half a gram of dry ice was sprinkled on the water and the jars resealed. Each measurement and dose was spread out by 1 minute to stay consistent. The pH of each jar was recorded at 30, 60, 90, and 120 minutes following dry ice addition. The second experiment tested water acidification with 3 doses of carbon dioxide. The jars were washed and prepared again. The pH of each jar was measured. Following the first 3 measurements, 0.5 grams of dry ice were sprinkled on the water and the jars resealed. The pH was then measured at 120 and 150 minutes without an addition of dry ice. The final experiment was similar, but dry ice was added after every measurement. For statistical analysis, I used a 2-way ANOVA test along with a Tukey test with an online calculator. Statistical significance was determined between groups. A 1-way student's t-test was used to determine if there was a statistically significant difference in means. This video demonstrates how dry ice was placed into the jars. The pH was recorded on a spreadsheet. There were 5 trials of 4 groups with 30-minute increments. The pH is also recorded at 150 minutes in the second and third experiment. The data collected was then put into graphs. Overall, there was a decline in pH with the addition of dry ice. During the first 30 minutes, all of the trials had a steep decrease in pH, except for the control in the continuous dosage experiment. The results of experiment 1 are shown in this graph. The ANOVA test indicated a statistically significant difference between groups at all time points. At the first time point, before dry ice was added, there is a discrepancy in pH between the 4 experimental groups, indicating the addition of lime in plants affected the pH. Both groups with lime were basic. At the second time point, after dry ice was added, there was a drop in pH in all samples. The jars with lime reached a neutral pH and had the steepest slope. The pH of the jars with only plants dropped as well. And even though they dropped to more acidic levels of pH, it wasn't as steep a slope. The control jars had a similar slope to the ones with just plants. The rest of the time points had a stable pH for all the groups. On the post hoc Tukey analysis, all the group comparisons were statistically significant except for the comparisons between the lime jars and the jars with both plants and lime, although the jars with both were consistently higher than with just lime throughout. There was also a marginal rise in the pH after the initial drop. In the control group, the pH trended upwards, but there was not a statistically significant difference between the 30-minute time point and the 120-minute time point. However, the pH increase in the plant group, lime group, and plant and lime group were statistically significant. The results of experiment 2 are shown in this graph. They were very similar to the results of the first experiment, but the rise upwards started at 90 minutes instead of 30. This was due to the dry ice being added at 30 and 60 minutes as well as 0 minutes. After the doses of dry ice ceased, pH trended up in the control group, but it was not statistically significant between the last dose of dry ice and the final measurement. However, the pH increased in the plant group, lime group, and plant and lime group between 90 and 150 minutes was statistically significant. The results of experiment 3 are shown in this graph. The results sloped downwards throughout this experiment as the dry ice was added throughout. At the time point 30 minutes into the test, the controls have a higher pH than the plant jars. As each experiment should be the same at this point, it may be an experimental error. The data shows that lime and java ferns prevented and reduced acidification of water following the addition of carbon dioxide. After dry ice was added, there was a drop in pH. With the addition of plants and lime, less acidification occurred and the pH rose after the final dose of dry ice. Neither were able to return the acidity back to normal at the dosage used, but they did minimize the acidification effects. They are then viable as a buffer in ecosystems from acidification by carbon emissions. The addition of lime made the water cloudy and may have changed the quality of the water. The cloudiness of the samples with lime may have also prevented the plants from performing photosynthesis. The plant would then take in less carbon dioxide. More acidification would ensue. We can then hypothesize that plants have the potential to reduce acidification in the right conditions. To further research solutions for acidification, longer and more testing would be needed. As the data had a slight upward slope after doses of carbon dioxide stopped, it would be interesting to see if the trend would continue given a longer testing period. In real ecosystems, there are other variables affecting organisms, so many water quality tests would be needed. Exploring the effects of lime on the plant's photosynthesis would also be a key step in finding a solution for acidification. It is difficult to compare two differently sized variables, so to clearly see if lime is truly better than plants, different amounts of plants, lime, and dry ice would need to be tested and compared. The current study is powered for the two variables used. Future studies would benefit from larger sample sizes. All in all, both plants and lime had a positive effect on the pH levels of water, so identifying effects of each is vital to prevent the acidification of bodies of water. I wish to acknowledge my mentors who assisted with experimental safety, statistics, funding, and editing. My mother helped handle the dry ice and provided the materials needed for experimentation. She also helped guide me towards the correct statistical analysis needed. I would also like to thank my science teacher for providing advice. These are the sources used to research my experiment, and these are the sources of the images throughout the slideshow. Thank you.